Okay, so I hope you're feeling refreshed, ready for a bit more brain food. Our next speaker is a much valued member of our board. Dr. John Broon has brought to our ACC&D family a suite of skills and knowledge from wildlife ecology that we have shamelessly exploited for the benefit of our mission. You can read more about him in the app, and I know you're really going to enjoy the insights he has to share with us today. So here's John.
Thank you. Fertility control tools, whether they are surgery or non-surgery, are, of course, applied at the level of the individual animal. But many of us, in applying those tools, hope to affect some sort of population-level outcome. It's certainly not the only reason, as we've heard from multiple speakers, why we may be interested in using these tools, but it's an important one. So, given that, the question that we face is, can a given fertility control tool be used to achieve a desired population level effect at an acceptable cost? That's our general question. Our specific question for this particular talk is whether or not a temporary contraceptive would represent a tool that could be used legitimately and practically to achieve a population level effect in a population of dogs or cats. The work that I'm going to talk about today and also tomorrow during the field track session is the culmination of a larger effort that's been undertaken by ACC&D to explore these types of questions about how to best use fertility control tools at the population level. But since we're only talking about a subset of those results today and in a relatively short time frame, only about 20 minutes, all of the rationale, the approach, the methodological points of discussion that have to do with this broader effort that we undertook at ACC&D is going to be deferred until tomorrow, so I have time to jump into some actual results for this particular facet of the study. If you are interested in those things, try to come tomorrow during the field track session, or if you're not able to do so, I'm always happy to talk about methodology after the presentation at some point. So just a little bit of a biologist's perspective on the difference between individuals and populations. It's obvious to many of you in the room, but it's surprising to me over the years how frequently this distinction sort of gets blurred or muddled. The illustration here just makes the point over on the left-hand side of the screen that we can know an awful lot about an ant, interesting little organisms, and about how they're structured, about how they physiologically operate, and have absolutely no ability to predict how they will behave in a swarm when they've caught a scent that represents food to them. Similarly, we can know an awful lot about how a cat functions physiologically, behaviorally, anatomically, and that tells us very little. It doesn't tell us enough about how a population of cats will function in order to be able to manage it as efficiently and effectively as we can. Populations of animals have various properties that are not properties of the individuals themselves. They exist at some sort of carrying capacity, which is a balance of sorts between those organisms and their environment. It determines how many of them can live in a particular place at a particular time. Fecund animals like cats can produce a lot more babies than will ever survive in the real world, meaning that it's not just a matter of them actually realizing their full reproductive potential in the real world. The rate at which individual cats survive or reproduce can vary depending on how dense the population of cats is within which they live. This is called density dependence. Populations will sometimes take a long time to realize or exhibit the effects of the management that we exert upon them in terms of lag times. And of course, most of these populations that we hope to manage aren't actually closed systems, but there are cats either leaving the system periodically or entering the system via immigration or abandonment. So all of these things are realities that we have to understand at some level if we hope to manage a population of cats with a given tool effectively and efficiently. The impetus for the work that ACC&D did on this topic was this paper by Levy et al. Doing a control test and a contained small population of cats in the laboratory using Gonacon, which has been discussed earlier. And what you see in this particular curve is a line that depicts how the proportion of effectively infertile cats changes over time. So over the first 6 months to a year, most of the cats who received Gonacon stayed effectively sterilized, right? Even though some of them started to revert to fertility quickly, most of them stayed infertile for the initial period represented by this curve. And it took, let's see if I can get this cursor to show up. Let's see if that's, I think, yeah, you guys see that in the middle? That's about the point at which roughly 50% of the cats remain contracepted and 50% are now again fertile. That occurs at about the 3-year mark, and the curve continues on out to the right. So the question we wanted to ask at ACC&D is that, okay, if this actually proves to be the case when Gonacon is used more broadly, which as an aside it turned out apparently not to be, but were that the case, is this a good tool for population management or not? So we assembled a group, a very diverse group, to investigate that question. You see the names listed here. Many of these names are people who are at this meeting or have been to previous symposia. Many of them are ACC&D board members or scientific advisors. But of these people, I would like to call out the one at the top, Dr. Phil Miller, who's with the Conservation Planning Specialist Group, who has a kind of a focal area of work that uses the methods that we employed for this study. Which was simulation modeling, which I'll talk about quite a bit more tomorrow. It's really the only practical way to investigate a question like this because we can't do experiments on these complex systems over and over and over again, practically speaking, within a reasonable frame of time. So these are our partners. ASPCA and Muriel were funders of this particular study. What I'd also like to point out about this group, which made the work, I think, particularly robust, is that it represents a wide range of expertise. We have people there who are cat experts, for lack of a better word, at the epidemiological or veterinary levels. We have folks there who have a very strong animal welfare perspective. We have pure-out biologists, some of who focus on the economics of population management. So we have a very diverse group here. Interestingly, the framework we use for our simulation modeling, which is called Vortex, illustrated down there in the bottom right, is predominantly used by Phil Miller and people like him to investigate the process of extinction and endangerment by certain wildlife species around the world. So we just turned it to our particular purpose here. So very quickly, we're jumping into some results here. The first question was, how does something like Gonacon compare, were we to use it in the real world, with our gold standard, if you will, which is surgical fertilization? Now, it stands to reason that something that's temporary won't be in some respects quite as good as something that's permanent, correct? So that's a bit of a given, but we'd like to come close and maybe have some cost advantages in the process of doing so. Now, I'm going to explain this a little bit so that it makes sense to you, so bear with me. There's 2 lines superimposed at the bottom. One of them's a blue line, and in the legend, it's labeled sterilized 75. That's our basic TNR scenario. And what those words and numbers mean is that in this model, every 6 months, we go and visit our cat population. We catch 75% of the cats who need to be sterilized, and we fix them, and we put them back out. 6 months later, we go out again. Some of the cats will already be sterilized, right? But some won't be, the ones we didn't catch the first time, plus the ones who showed up during that intermediate 6 months. So 75% of the ones who need to be sterilized at that point, we catch and sterilize. So that's what these numbers mean. And that curve that you see down at the bottom is where we end up after 10 years if we do that every 6 months over and over and over again. So we're calling that sort of our gold standard under these circumstances that we modeled. And there's 2 things to point out here about how contraception compares to that. The 3 contraception curves, which have these kind of gibberishy sounding labels in the legend, are just different levels of intensity of applying a contraceptive like Gonacon. The curve at the very bottom, which is superimposed on the TNR curve, is one in which we do the following. We contracept every single cat the first time we go out. When we go back 6 months later, we contracept all of any new cats that have showed up. And then after a year, we've got a whole bunch of cats that were contracepted a year ago, and we do them all over again. And we keep that process cycling. So we're basically doing our very best in that curve to keep every single cat contracepted at a 1-year retreatment interval, which is not very practical, really, when you think about it. But that's what it takes to get the same population level result as surgical sterilization. And as we decrease the intensity of the contraceptive effort, which these successive upward moving curves do, we get a less good population level outcome at the end of 10 years. So that's a little bit sobering, not surprising perhaps. The reason, of course, is that with a temporary contraceptive, we have to retreat animals periodically in order to keep them from reproducing. And this is just a very messy looking graph that shows you over time what that looks like and how big the difference is in the number of cats that need to be treated under these different scenarios. You can see the blue curve down at the bottom. I don't see my curve. There it goes. My blue curve down at the bottom, which is sterilization with TNR. The number of cats we have to treat declines pretty quickly over time and stays fairly low. With a contraceptive scenario, we have to keep retreating cats as time goes by, so the number we have to treat cumulatively over time is pretty high. And the consequence of that is that, somewhat to our surprise, the cost of achieving these outcomes at a population level with a sterilant like Gonacon, given the admittedly uncertain assumptions we made about pricing, is not only not cheaper than doing sterilization, but quite possibly more expensive. And that's because of the retrapping effort that's required and because of the fact that many individual cats are treated multiple times during their lifetime. So this is kind of sobering, at least at this point in the analysis, although that's not the end of the story. And so we asked the question, we reframed the question to this: how much better than Gonacon, as we then understood it, does a temporary contraceptive have to be before it starts to offer compelling advantages compared to surgery? Now, there are several ways that are not probably as complicated as they're going to look in these slides that we can envision for making a contraceptive that's temporary better. For the successive graphs here, I'm going to suggest those little inset graphs in the upper left we just ignore for the sake of time and concentrate on the bigger graph. And what we see here is a red line, which is the topmost line in this graph. That's just the reverse of the curve that was in the Levy paper. Instead of showing how cats go from being all contraceptive to all fertile over time, this shows how the percentage of cats that are fertile rises from the beginning when no cats are fertile because they've all been contraceptive, over the course of, let's say, up to 3 years, when about half of them have returned to fertility. And even after 6 years, only about 85 or 90% of them have returned to fertility. That's Gonacon. But how could we make that better? Well, one way we can make it better is by reducing the slope of this line, like we see here and here and here. So if we look at this line, for instance, we still have cats returning to fertility from day one, but they're returning at a slower rate. And at that 3-year period, only about 20% of them are fertile, right? That's one way we could improve a temporary contraceptive. We call that slope modification. A different way to improve it would be what we called intercept modification. In that, the slope at which a return to fertility occurs stays constant, but the time at which it starts changes. So in this first curve to the right of the red line, what we have is essentially the Gonacon curve, but it doesn't start invoking itself until 6 months in. And way out here at the right, we have a similar return to fertility curve as Gonacon, but it doesn't even start to invoke itself until 3.5 years in. That's another way to improve a contraceptive, temporary contraceptive. A third way would be something that we called, for reasons that have been lost in the historical winds of time, is called trade-off modification. In that particular set of scenarios, we keep the mean duration of the contraceptive constant at about 3.5 years, but we compress the time frame over which that return to fertility occurs. So here we have, oops, let me go back to the one I had, sorry. Here, the red line is what we saw before, but the rightmost line, my cursor's not working, sorry. The rightmost line still has a mean return to fertility of 50% at 3.5 years, but the whole process starts in year 3, as you see, and is completed by year 3.5. So it's a very narrow window, almost a deterministic return to fertility. And then finally, there's sort of a mashup of that, which you see here, combined modification, in which we reduce the slope of return to fertility and at the same time increase the length of time before that return to fertility curve actually starts. Now, I showed you about probably 40 different lines on those previous graphs. It wouldn't be fair to show you the results for all 40 lines, so we're just going to take a sample from each of those previous 5 or 6 slides to actually look at. So what you see here, again, you see the red line, that's the Gonacon return to fertility, and then you see one line from each of those previous 5 slides, kind of in the middle of the range that we tested. Those are the ones that I'm going to show you some results for here, and for the sake of comparison, we invoked all of these new hypothetical contraceptives at a sort of intermediate level of application intensity. So instead of trying to contracept every single cat, we contracepted 75% of the ones who were potential targets at 2-year retreatment intervals. And what we got when we did that was some better results, not surprisingly. The results, and don't worry about what they are because I'm going to show you again in just a second, but 3 of our improvements resulted in a population level outcome that was basically as good as our TNR outcome. So that's encouraging. 3 of them didn't. Hold that thought for just a second. Again, remembering that all of our pricing information is fairly conjectural, we see some incremental improvements in cumulative treatment cost with these moderate improvements in contraceptive as we go out to columns 3, 4, and 5 of this graph. They're not pronounced, but they're moderate, perhaps encouraging. But here's where we really draw our conclusions. These, again, are the 6 different scenarios that we're comparing. We're comparing Gonacon, the red line, to these 5 different flavors of improving contraceptive efficiency. Now, the lines that are marked with the yellow arrows are the ones that produced as good an outcome as surgical sterilization at a more or less comparable costs. And if you look at those 3 lines, the slopes are very, very different, right? And the top line with the yellow arrow, the mean efficacy is about 3.5 years. In the bottommost line that has a yellow arrow, the mean efficacy is, it looks like it's going to be 7 or 8 years. We don't even get up to 50% at the end of the x-axis. But what they have in common is that they all give us 1.5 years before any cats start returning to fertility. Those are the lines that are improvement over Gonacon. The lines that all have an intercept at or near 0, meaning that cats start to return to fertility immediately at whatever pace, are all not as good. So we have actually a pretty clear lesson here, I think. And that is, well, there are several. One is that the parameters under which a temporary contraceptive starts to make sense as a population control tool are perhaps a little bit narrower than we thought they would be when we started this exercise. And the key factors that distinguish a hypothetical temporary contraceptive that's good for population control from one that's not so good, as far as we can tell through this exercise, is that it offers us a very clear period of, there's not a definitive time frame, but some period, a half a year, a year and a half, maybe 2, when we're sure or almost sure we're going to get full contraception or full fertility control for the cats to which that's been applied. The anathema to using these temporary contraceptives for effective fertility or population control is that immediate return to fertility at whatever rate it starts to happen. Another factor is that because we have to retreat so many cats in a contraceptive scenario, we can control costs particularly well if we're able to subsidize a lot of our cat capturing effort through volunteers without having to pay for it overtly. That said, remember we go back now, and for a temporary contraceptive to make sense or to be usable, it doesn't necessarily have to be better in all respects than surgical contraception. There might be logistical reasons why we're happy with a temporary contraceptive that's as good, financially speaking. For instance, we're working in a part of the world where you just cannot get enough high-quality, high-volume veterinarians to do surgical contraceptive. So if we had a temporary contraceptive that does as well, it means it can at least be used in a broader range of places, right? Also, if we're talking about dogs, we've got a little bit different situation. Now, we don't know if these results apply to dogs to the extent that we do with cats, but let's say they do for the sake of argument. Dogs are, in my experience, at least in many places, potentially easier to get your hands on to give a temporary contraceptive to than cats are. Cats require considerable effort, or they require notable effort, in some cases considerable effort, to get a hold of. Many dogs also come through rabies control programs on regular pulses every year, every 2 years, however often. So there are kind of leveraged advantages of a reasonably effective temporary contraceptive for dog population control that may not be so pronounced in cat populations. So with all that in mind, we have sort of a mixed set of results, but there is an answer, I think, to how long is long enough for a temporary contraceptive to start to become useful as a population control tool. And we are planning to probably do a little bit more work to quantitatively define what those bounds are under a set of different circumstances or a set of different financial constraints. So with that, I think I'm more or less on time. If you're interested in this broader set of work, tomorrow we're going to be discussing it at more length with a broader set of questions at play. Thank you.